Okay, welcome to OsteoBites, everybody. My name is Anne Graham. I'm an osteo warrior and executive director of MIB Agents. It's a gorgeous fall day. So glad you're joining us. It's also a very exciting fall day for osteosarcoma because we have a new clinical trial to talk about. This is what we wait for. This is what we're like, let's go. We need this to work. This one is for relapsed refractory osteosarcoma. Not only that, we get to talk with two very smart and very cool doctors who are dedicated to better for our kids with osteosarcoma, Dr. Glade Bender and Dr. Filemon Dela Cruz. Dr. Glade Bender, you may be familiar with from our collaborative MIB Agents, Hyundai Hope on Wheels Thought Leaders Conference and our MIB Factor Conference. She received her MD from University of Pennsylvania and practices at Memorial Sloan Kettering Cancer Center, kind of my alma mater. She is the vice chair for clinical research and is a pediatric oncologist who specializes in caring for young people with solid tumors of the bone and soft tissues, including osteosarcoma, Ewing sarcoma, rhabdomyosarcoma, all the usual suspects that we need to start defeating solidly. Dr. Dela Cruz received his MD from UC Davis, and he is a pediatric oncologist who also cares for children with sarcomas, including Ewing sarcoma, osteosarcoma, rhabdomyosarcoma. He is actively involved in the pediatric translational medicine program at Memorial Sloan Kettering Cancer Center, where they're using new technologies to gather information about biology and genetics of each patient's tumor to help guide treatment decisions that specifically target an individual's cancer. So here for that, really exciting work. Also, he's involved with leading clinical trials of new treatments for cancer, specifically pediatric sarcomas like osteosarcoma. Before we get started with this conversation today that is relevant to so many of us, please allow me to tell you that MIB Agents makes it better, MIB, for kids with osteosarcoma through our programs, research, and education. Our education programs include our annual MIB Factor Conference, our testing and research directory, which helps inform a personal treatment plan or osteosarcoma research, OsteoBites, our clinical trials 60-second search, which lives on our website, and our book, which I happen to have here, Osteosarcoma from Our Families to Yours, which was also worked on by Dr. Dela Cruz's and Dr. Glade Bender's colleagues, who are also on our board. And the second thing is our MIB Agents support programs. This includes a verified osteosarcoma resource packet, which you can request. We send it out free of charge. It's full of resources, not only from MIB, but from other supportive organizations that help osteosarcoma patients. We also have a gamer agent program, warrior mail, ambassador agents who are trained to support their fellow osteo warriors. We have prayer agents, end-of-life missions, bereavement and young adult support group for survivors. Finally, research. Our Outsmarting Osteosarcoma funds osteosarcoma specific research, supports researchers, and we share that research as much as we can. We love osteosarcoma researchers so much. No charge ever to participate in MIB Agents program. Therefore, we need your support on Giving Tuesday. We are committed to raising $43,000 for the 43 kids who are diagnosed with cancer every single day. Together, we can cause a cure. Not kidding. So please remember us on Giving Tuesday. Okay, that's my long introduction. Dr. Glade Bender, would you introduce yourself, please?
Hi, I'm Glade Bender, and I'm thrilled and delighted to be able to join OsteoBites today. I, as Anne said, am the Vice Chair for Clinical Research at Memorial Sloan Kettering and also a member of our sarcoma service. and really have spent most of my career doing early phase clinical trials, new agent research for children with relapsed and refractory cancer. So today's session is really going to focus not only on a single trial, but sort of the landscape of what is out there right now for relapsed and refractory osteosarcoma. And I'm really here to support my colleague and friend, Dr. Filemon Dela Cruz, who is really one of the spectacular translational researchers who not only helps with the clinical trials, but also does all of the preclinical research in the lab to support their development, to make sure that we're really only bringing the most promising therapies into the clinic for children. So Phil, I pass it to you.
Great, thanks, Julia. So I'm Phil Dela Cruz. I also am a sarcoma doc here. But what you might not know is, yes, I've known Julia for a long time, and she was and continues to be my mentor. So we knew each other when we first met at Columbia before we came over from Memorial. But yeah, as Julia said, this was, sort of my involvement and sort of my first job out of college actually was in a laboratory that happened to do research in, amongst other things, sarcomas. And it was so very interesting and very exciting actually to come back, more than a decade later to sort of see what happened since. And, I think a lot of the things and a lot of the trials that we'll be sort of talking about today are, I think it really is built upon, an incredible amount of work by so many people. And so it's very sort of humbling, and, it's always sort of a privilege to work, in this field, and I think osteosarcoma is something that, I know we all feel very passionately about, because as I'll show shortly, there are some things about it that are still quite frustrating in terms of, changing the outcomes for patients with osteosarcoma. So, but as Julia mentioned, I also, in addition to seeing patients, which was my first love in medicine, I also sort of have the privilege of going into the laboratory and learning more about, yes, what treatments are effective, but also just as important, because believe it or not, a lot of the treatments that we use, we actually don't know how they work, or they don't know who they work better on. And I think that's one of the challenges. And so that's one of the things that, myself and many others, who do this sort of work are sort of motivated by. So thank you for, this is exciting. So thank you for the invitation to speak with you guys today.
My pleasure. Anna, go ahead.
I'm Anna Eggleton, and I'm very proud to be MIB Agents Director of Marketing, and just very excited to be here with these awesome researchers and scientists.
So originally, when we were planning this talk, we had originally thought we would just be talking about one clinical trial, and we will still talk about that one trial, specifically the OLIE clinical trial, but things have changed actually fairly recently, which is why we thought to sort of expand the talk and talk about, as Dr. Glade Bender was saying, sort of the landscape of other studies, other clinical trials, both sort of that are open, will open, and then in the end, I want to talk about sort of things that aren't quite at the stage of the clinical trials, but hopefully may sort of enter the stage for patients with relapsed refractory osteosarcoma. And this is sort of our standard disclosures, but I think none of the sort of our disclosures and involvements have, have any direct effect on the content of what I'm about to discuss. And so I always like to start with sort of like general and the basics. So a lot of us know all about osteosarcoma, but as you can see in terms of, what is the proportion of patients with osteosarcoma compared to children with cancer, it's a minority. So bone tumors make up about 5%, but within this, osteosarcoma is the most common malignant tumor of bone that is that occurs in children and in young adults. And it also has this bimodal distribution. So the majority of the patients that develop osteosarcoma tend to be individuals who are in their sort of adolescent years and young adults. And then there's a second peak that happens much later on in life and is associated with Paget's disease of the bone. And this sort of are the survival statistics. These are the outcomes of patients who are diagnosed with localized osteosarcoma. And what these generally sort of show is that for the most part, patients with localized disease, about 2/3 of them are able to be cured of their disease. And you see this big jump here, and this big jump happened because of the use of systemic chemotherapy in the treatment of osteosarcoma. But as you notice, though, that it doesn't keep going up. It sort of has plateaued in both situations. And I think the thing that really motivates us is this graph, which is probably one of the most sort of frustrating graphs to look at, which is sort of the trend of survival of patients who have metastatic disease when they present. And this is very similar to patients who have their disease relapse as well. And what's pretty sobering about this is the fact that despite many decades of treating this disease, we've not really made a significant sort of impact or improvement in this group of patients. And this sort of is the results and the outcomes from sort of our last large study in osteosarcoma that looked at the standard 3-drug or the MAP chemotherapy and asked the question, well, if we just add more chemotherapy that also seems to be active in osteosarcoma, specifically this IE or ifosfamide etoposide, do we improve the outcomes? And unfortunately, the answer to that was not really. So, and so we're still sort of stuck with the, and in terms of how well we're able to cure patients with disease. And again, here's that really, sort of sad or sort of, outcomes in terms of what happens if you have metastatic disease when you first present to the oncologist. Again, doesn't seem to, adding more chemotherapy does not seem to significantly affect the outcome of patients. And so what do we do? So we know that, chemotherapy works, but we know that we really need to think about doing something different and adding something different that works sort of in a different way than how chemotherapy works. And so now we're sort of, now you've heard the term precision medicine and the term of molecular medicine. And so in the past decade now, there have been sort of these newer drugs, or these so-called small molecule inhibitors that have been evaluated in cancer, including cancers like osteosarcoma. And one strategy was, this thought of targeting this process called angiogenesis, which is the process of creating a blood supply. So just like your organs need oxygen and nutrients to sustain itself, so does a tumor cell. But a tumor cell shouldn't be in your body, but it sort of is able to feed itself because it has this ability to create its own blood supply. And again, this process is mediated by a variety of proteins, or this process of angiogenesis is mediated by a variety of proteins, including this protein called VEGF, which stands for vascular endothelial growth factor. And tumors can secrete this or the receptor to this protein called VEGF receptor to establish a blood supply and allow it to grow. And so the thought of, what if we can then target or prevent a tumor cell from occurring, perhaps we can then sort of starve and as a result, kill the tumor cell by using drugs that have now been collectively referred to as anti-angiogenics to sort of suffocate the tumor cell. And so we're able to accomplish this by these small molecule drugs that are some of which are called tyrosine kinase inhibitors or TKIs, that are small molecules that will either bind to this VEGF protein or the receptor of VEGF here and prevent their interaction, because when they interact, then that results in a cascade of signals that allow a tumor cell to start creating these blood vessels. And some of these drugs, as you can see in this picture, are, include now really a variety of drugs. A lot of these drugs end with IB, and I think some of these you might also be familiar with. So things like sorafenib and pazopanib, both of which have been used in osteosarcoma and other sarcomas with varying degrees of success. And so we know in osteosarcoma that just like with other tumors and other sarcomas, that we see elevated levels of VEGF, and which is one of the things that tip people off in terms of using this approach or this strategy to try to treat this type of tumor. And we know that this is also linked to their ability to travel or metastasize to other sites. So, these tyrosine kinase inhibitors, these small molecule inhibitors have been around now for many years. And then there are sort of newer generations that have been developed that have sort of different degrees of selectivity in terms of what they can bind to and inhibit. And so the idea of why don't we take these sort of newer generation agents and combine them with something that we know is also effective and has activity and can kill osteosarcoma, meaning chemotherapy. So things like MAP or things like ifosfamide etoposide. And so that was sort of the basis and the rationale for, trials like OLIE. What if we take, a small molecule and combine it with chemotherapy? Does that work sort of better? And hopefully we see something, or we get a result different than what we saw when we combined just a bunch of chemotherapy drugs together. And as I mentioned, sort of over the years, there have been many different small molecule or TKIs that have been developed. Sort of a newer generation TKI that was used in the OLIE study is this tyrosine kinase inhibitor called lenvatinib. And lenvatinib inhibits your VEGF receptors and actually all the different types of VEGF receptor proteins. But in addition to that, it also, and this is sometimes a good thing, it's not so selective that it just turns this off, but it also binds to and inhibits other proteins or other receptors, in this case, something called FGFR, PDGFR, and RET. These are just other proteins very similar to VEGF that are involved in allowing a cell to proliferate and also to metastasize. And so with this particular agent also, there are, before you do any sort of studies in people, there are a lot of studies that are done in tumor models in the laboratory, including models of osteosarcoma. So this drug was also combined with chemotherapy in a mouse model of osteosarcoma, and it was shown that the combination caused the tumor to regress much more so than when you give either one by itself. And so that was sort of the motivation or the rationale for now exploring this combination in patients with osteosarcoma. And so this was actually the predecessor study to the OLIE trial, which is the study 207 that was led by Dr. Nathalie Gaspard in France, as well as the rest of the European ITCC group that looked at initially in the safety. You always want to make sure you know that when you combine this and give it to people that it's safe and it's tolerable. And if so, what is the dose that we should use? And so that's one of the primary purposes of this study. And they looked at a population of patients who have osteosarcoma, who had either relapsed or who had progressive or refractory disease. So these are patients who have all gotten standard chemotherapy, likely MAP chemotherapy. And so they needed something else. And these patients tended to have lung as their most frequent site of metastasis. So this is sort of a almost typical osteosarcoma patient who relapses, or were the patients that were treated in this study. And so the results of this study was published, and they did find an effective or a tolerated dose of the TKI, of the lenvatinib, in combination with ifosfamide etoposide, which is given once a day for 3 days, every 21 days. And what they also found in this study was actually preliminarily that there was, in addition to this being sort of a tolerated dose, the patients who were treated with a combination had a significantly improved disease progression outcome at 4 months, meaning at 4 months, what is the likelihood that a patient's tumor the likelihood that the tumor had progressed at that point. And that was found to be 51% at 4 months, compared to the historical control, compared to what if you don't do anything, which is 12%. So this is a very promising signal. And the side effects that were observed were not any different than what we would expect with any of these agents. And the predominant side effects that were observed were myelosuppression, or decreases in the red blood cell, white blood cell, and platelet counts, as well as nausea and vomiting. And so because of the promising sort of outcomes that was seen in this study, that was the basis ultimately for OLIE Study, which is study 230, which is really a confirmatory study looking at this combination of lenvatinib plus the chemotherapy, ifosfamide etoposide. And the goal was to treat 72 patients. And these patients were randomized into these two groups. So either the combination group, or arm A, or patients who just got the standard chemotherapy, which was ifosfamide etoposide. And in this study, patients who were randomized to just getting chemotherapy, if they actually progressed, there was the option to cross over, which meant these patients could then continue to receive the chemotherapy, but now add on lenvatinib, just so that they would have access and to see if they may have benefit by adding the TKI on top of it. And so this is the aim, similar to this is sort of the measure with which we are going to look at to see if this indeed is something that is truly active in patients with relapsed refractory osteosarcoma. And that's, again, looking at sort of this since we have a benchmark at 4 months for these patients, looking at whether or not they progress on therapy at 4 months. This study was an international study, and I think this is a really important point. It's to do these types of studies in a very rare disease or tumor type like osteosarcoma. But one of the challenges of these types of studies is getting enough patients to answer the question. And really the only way to do that is to sort of band together, and that is sort of what one of the sort of wonderful things about the OLIE study was this was an international collaboration. Actually, just like the largest sort of COG study in osteosarcoma, that was also that evolved into an international sort of collaboration because we knew it was just taking so long to get enough patients to answer these really important fundamental questions. And because of that, and why we've sort of retooled this talk is because of this, we have been able to actually complete accrual onto this study really just in the past month or so. So now the patients, so we have the patients and they're being treated and really now they're being monitored. So now we sort of wait to see if the activity that we were seeing in the earlier studies sort of bears out. So I think we'll hear more about sort of the outcome of this study in the next several months or a year. But I think we're forever optimists, and so hopefully we actually see an improvement and see something that just we haven't seen in such a very long time. And so, this wasn't actually the very first time we've looked at these TKIs before. I think a lot of us here are familiar with the TKIs and sort of their role in osteosarcoma. I think a lot of people are familiar with this drug called regorafenib, which is also a TKI. It sort of is very similar to lenvatinib. It just has slightly different targets, but a lot of the same targets that lenvatinib had. And this was the study in relapsed refractory osteosarcoma that was done by the European group that was called the Weone study. And in this study, they saw it at 2 months, a good number of the patients that were not progressing. And there was actually a similar study that was done by the American group led by Lara Davis that essentially saw sort of the same thing, that treatment with just regorafenib by itself, and this is an oral TKI, did improve or lengthen the progression-free survival by a couple of months. So not a home run, but it's just we want to take sort of these small sort of successes and sort of use them and develop on top of them. And then most recently, another TKI, something called cabozantinib, in the K-bone study also had sort of fairly sort of similar type of result in terms of being able to at least stabilize and slow down the progression in patients with relapsed osteosarcoma. So what other trials? So what's sort of been exciting for osteosarcoma, at least from our perspective, is, we were very frustrated because for a long time it was just regorafenib and cabozantinib most recently, but there really weren't a lot of other clinical trials for osteosarcoma. But it really in the past year, all of a sudden, we're excited because now, and what I'm about to sort of describe are sort of the recently opened clinical trials for relapsed refractory osteosarcoma. One of them is this trial, which is conducted through the Children's Oncology Group, looking at this compound called DS8201. And I think there was a prior OsteoBites where I think Dr. Richard Gorelick talked about these classes of drugs called antibody drug conjugates. And that's what this DS8201 is. And what it is it's an antibody that recognizes a protein on the surface of a tumor. In this case, HER2 is the protein. And HER2 is a protein that's expressed in different tumor types. Most people are familiar with HER2 and its a role in breast cancer, but osteosarcoma also expresses HER2 on its surface. And so we can sort of capitalize on that fact, and that's what the antibody does. But all the antibody does is it directs the payload, and the payload is, in this case, chemotherapy. So it's a way of sort of concentrating and directing your treatment just to the tumor cells with the hope that you have less collateral damage in the form of, less side effects or lesser side effects than what you would see if you just gave chemotherapy. And so that's what this is. This is a conjugate or a combination of this antibody linked to this chemotherapy. And this is sort of studies that were done also by our group. Other groups have also done this, including Dr. Richard Gorlick's group. But the point here is that, just with a couple of doses of this, DS8201A, it's also called trastuzumab deruxtecan, we were able to see these are osteosarcoma tumor models in mice that were generated from patients. And what you could see here, and what I hadn't seen in a very long or ever, are regressions in osteosarcoma. So this and work by other groups was what motivated and provided the rationale for this phase 2 clinical trial in patients with osteosarcoma whose tumors also have to express or have HER2 on its surface. And the patients must have measurable disease or something that you can measure on a scan. As you see here, this trial, while it's open, is currently on hold because they have accrued and treated sort of the first set of patients. And so when that happens, the study sort of goes on pause as we sort of evaluate the outcome on those patients, and also to sort of confirm that this is safe and patients who are being treated are not getting sort of or experiencing unanticipated side effects. So this is sort of one sort of new class of agents that are being used in osteosarcoma. The next one, also conducted to the Children's Oncology Group, this is led by Dr. Damon Reed, is this agent called OST31164. I don't know if it has another name, but this is a type of an immunotherapy sort of trial. And this is based actually on a very promising work in dog or canine models of osteosarcoma, where this agent actually showed pretty good activity. And so just very generally, so what this is this compound essentially is infused into a person, and it says here it's a type of a vaccine, but essentially you're infusing sort of the instructions for a chimeric protein, so a hybrid protein of HER2. And what happens when you infuse this into someone, it's taken up by these immune cells called antigen presenting cells, which then express this sort of protein on this HER2 hybrid protein on its surface. And what happens when that occurs is that you have these other tumor cells in your body, these things called CTLs or cytotoxic T lymphocytes, that then get trained to say, okay, here's HER2. I'm going to go look for HER2 at other parts of the body and kill them. So this is an immune-based strategy. And as I mentioned before, osteosarcoma has HER2 on its surface. And also as a byproduct of this, there are cells that are normal immune cells that are present in our bodies that suppress immune responses. And so this agent is also somehow able to sort of inhibit the immune process that prevent our immune system to identify tumors. Tumors should not belong in our bodies, but unfortunately tumors have evolved ways to blind our immune system to their presence. And so this is, again, I believe they've have accrued a patient at least onto this study. So this is very exciting. This has also been many years sort of in development. So it's very exciting to finally see it happen. One thing with this compared to the prior trial, patients must have no evidence of disease to be eligible for this. So patients who have relapsed have to have their disease sort of taken out surgically, and on scans can't have any disease to be eligible for this trial. And then the next study is a study actually that is being led by Dr. Katie Janeway at Dana-Farber. And this is looking at these two drugs. One is called olaparib and the other is ceralasertib. And what are these things? And so this is this is sort of what normally happens in a cell. So a cell, before it divides, has to make more copies of its DNA. But invariably, what happens over time is your cell can make mistakes as it's making or replicating its DNA. That normally happens. And our cell has normal processes to say, oh, okay, if you have damaged your DNA or a mistake happens, there are proteins like a protein called ATR that can pause sort of this process, pause it so that the cell can then go and fix the mistake that happens or fix any damage. And so these are these things called checkpoint. And you may have also heard of these classes of drugs called checkpoint inhibitors. But this is sort of that checkpoint, and a cell has different checkpoints before it is able to divide into two cells. And so, and tumors, as you can imagine, have figured out how to use these proteins to its advantage. Related to this is another protein called PARP, which if you have a mistake in your DNA, you need a way to repair it. And PARP is a protein that's involved in repairing any damage that happens to your DNA. So then why would you want to combine the two of them? Well, if you think about it, and what happens in tumors like osteosarcoma that can increase and jack up the levels of ATR, for example, in their tumors, and this has been studied and has been associated with poor response to chemotherapy or relapse. If you block then with an inhibitor, in this case ceralasertib, which blocks ATR, you'd no longer get a pause. And so any sort of mistakes that you've accumulated or damage to the DNA that's accumulated will keep right on going, and it's going to keep trying to replicate itself. And then you hit it a second time and to prevent its ability to repair itself. And so what happens is you just get an accumulation of DNA. And then what happens when your DNA is really quite damaged is the cell is now unable, the tumor cell is now unable to replicate, and processes like something called apoptosis is triggered and the cell dies. And so that is the rationale behind combining sort of these two agents and evaluating this combination in patients with recurrent osteosarcoma. And there are two groups here. They are looking at patients with measurable disease, but also patients who are able to have all of their disease resected. And sort of what they're looking at, they're sort of will analyze these groups separately. The ones who are treated with measurable disease will have sort of that 4-month evaluation point as with the other studies. And then the patients who don't have any disease and then start on this treatment, we're going to ask the question, well, at 12 months, do they still have disease or not? And what is the proportion of patients who, despite having, surgery to remove all of their tumors and, being on this combination, what's the likelihood that they will have their disease come back despite all of this? So this is another study that's open at Dana-Farber, as well as our center and others. And then this is a related sort of mechanism. This is another trial in looking at this drug called ZNC3, which is a Weone inhibitor. And again, the same idea is if you inhibit Weone and you give chemotherapy, that will damage the DNA. And Weone is also involved in sort of trafficking and telling a cell to stop because you have damaged DNA, and essentially removing the brakes and causing something called, I love the term mitotic catastrophe. You're trying to divide, but your DNA is so messed up. The only end result of that is death of the cell. And this was a combination of this gemcitabine, which is the chemotherapy, and the Weone inhibitor ZNC3 was also evaluated in a mouse osteosarcoma model. And what you see here in the red is its ability to really slow down the growth of the tumor compared to this is just the Weone inhibitor by itself, and this is the chemotherapy by itself. So this is things that we like to see. This is also now a trial that's open in patients with osteosarcoma. And then lastly, this is a trial actually that I sort of run and I've inherited, and this is a phase 2 trial of Nexidimab, which is an antibody that recognizes a protein that's also expressed in a large number of osteosarcoma. In this case, this is the protein is called GD2, and it's given in combination with this growth factor, GM-CSF, which is granulocyte macrophage colony stimulating factor. And the idea is you infuse the antibody and it binds to the GD2 protein, which is present on the surface of osteosarcoma cells. And then you have GM-CSF, which stimulates the immune system and specifically stimulates the cells that are involved in identifying cells that are tagged with antibodies like this and eats them or kills them. And this is for patients also who have had all of their disease resected by surgery. And we are, this is a study that's been going for several years, but in the past year is finally opened at other sites, including Children's Hospital Los Angeles and MD Anderson. So hopefully by having additional sites able to offer this trial, we can finally complete accrual of the 39 patients onto this trial. And again, looking at disease control at 12 months. And so those are sort of those trials are sort of trials that are sort of highlighted just based on sort of the different mechanisms and the different rationales for them. And it was does not mean that that's all that there is. There's many other sort of ongoing clinical trials looking at different combinations of TKIs plus or minus chemotherapy. But really what's exciting, and I'm also sort of involved in evaluating other agents that, at least in the laboratory, seem to have activity for osteosarcoma. Again, there's now sort of a whole host of these antibody drug conjugates, or these strategies of delivering very potent chemotherapy that you normally couldn't give by itself because you would probably kill the person because it's so toxic, but delivering it in a very directed manner, but using proteins that are normally present hopefully just on the tumor cell, so that we selectively just bring the chemotherapy drug to the tumor. So and this is sort of an area that I'm also very excited in and very much actively exploring in the laboratory. And then there are these other sort of other strategies. There are these things called transcriptional inhibitors, which have also been shown to be active in the laboratory in osteosarcoma. I think we've all heard of these CAR T cells, same idea, sort of using the immune system or training the immune system to identify the tumor cell, but helping them along by giving them a target that's present on the osteosarcoma cell. And then, we've really only starting to study sort of the role of metabolism and its role in promoting cancer. And so there are these metabolic inhibitors that are being explored. These are things that inhibit the production of amino acids in the body. But there has been some work in primarily canine models looking at inhibitors of metabolism and combining them with things like metformin, for example, and at least early on in the laboratories, there seemed to be some activity in osteosarcoma. And then these other strategies that I sort of alluded to, and there are these inhibitors to this CD47 and SERP alpha. And the general idea behind these is, as I mentioned before, tumors have evolved ways to blind our immune system to them. And this is one of the ways that they do this. And so if we can inhibit that, we can then unblind our immune system with the hope that they can now see the tumor cell and eliminate them. And then lastly, these epigenetic modifying agents, specifically these drugs called HDAC inhibitors. And these are you can get a tumor not because you have a mutation or mistake in a gene, but how those genes are and to what level those genes are expressed are regulated by sort of the epigenetics of a cell. And so by sort of fine tuning and sort of getting things so that certain proteins are really only on or produced at any given time can be accomplished with things like HDAC inhibitors. And by modulating that, that there's also been quite a bit of work now in osteosarcoma looking at these epigenetic modifying agents. So a lot of very sort of things on the horizon, a lot more, I hope, sort of than what existed even a couple of years ago, which I think is really hopeful and really, good news for our patients. And so likely at the end, I'd just like to thank Dr. Natalie Gaspar and the ITCC group and ASI who sponsored the OLIE trial, as well as Dr. Katie Janeway and Dr. Damon Reed for all of their work and sort of their leadership and sort of these next generation of clinical trials. And then all of you and all of the patients' families. And lastly, Anne and Anna with MIB Agents for sort of giving us this opportunity in this forum to sort of to talk about these things, but also to sort of describe that, while there's still much to do, at least we have a lot more than we had, again, not too long ago for these patients. So I think with that, can chat.
Thank God for that. I mean, we really have had no place to go but up on options and what more can we do, especially when you have relapsed refractory disease. Just a quick question / confirmation. I believe the OLIE study is fully accrued. Is that right?
Correct. Yes.
So what does that really mean in terms of will more spaces open up can you explain what that process is like now?
Yeah, so, every study has, we say, an accrual goal. So you need X number of patients to answer whatever the question is for a trial. In this case, the OLIE trial has preset that they needed to treat a total of 72 patients. And after that, it stops. And then essentially at that point, they have enough patients in the study being treated or has been treated in the study to answer their question. And so they have the 72 patients. And so at least in terms of looking, of getting these agents within the context of that trial, that's now sort of done.
But maybe I can give a little more on that. These trials are really to shine a light on the way forward, right? And that's the point of them. So I think, first of all, Dr. Dela Cruz is being quite humble because the reason that he's presenting the OLIE trial is because while the OLIE trial, the trial of the lenvatinib with the chemotherapy was going on, he was also looking at a trial to combine lenvatinib with another targeted agent in the downstream pathway. So he was looking at the receptor and then blocking something down inside the cell to turn it off, something called everolimus. But they specifically told us when we were doing that study that we could not do osteosarcoma on that study because they were really looking forward to doing this combination in osteosarcoma. And that's because this is the most common chemotherapy used for patients with relapsed refractory osteosarcoma, and it works for some, but not for all. And while it worked for a period of time, it didn't seem to be durable. And so really trying to better on what we had that was best with the goal of, well, if this works, well, why wouldn't you move the tyrosine kinase inhibitor, the VEGF, and other growth pathway inhibitor to the beginning, up front in the brand newly diagnosed patients. And so the process of all of our clinical trials really take this stepwise process. And what the OLIE trial represents is sort of the hardest step, the second step, right? The first step is, can we give it to patients? Is it safe? Do we know what dose to give it at? And is there a signal that it actually looks interesting? Then the next question is, okay, well, what if we put it in the context of something we already use? Does it make it better, right? And if it does, then the idea is to bring it forward in newly diagnosed patients. And so the reason to bring up sort of all of the different multi-tyrosine kinase inhibitors, so the lenvatinib, the cabozantinib, the regorafenib, is that I think they all have a similar signal. Lenvatinib seems it may be easier to combine with chemotherapy. We don't know because we really haven't combined the others with chemotherapy, but that really is the goal that the COG is working on right now, is to see if we can design a trial where we're using one of these multi-tyrosine kinases together with MAP chemotherapy. And that would be the last step, the step 3, which is obviously the hardest because you want it to be safe. There's no guarantee that it's better than what we already have. That's why we do the study, right? We're trying to make it better, but we don't, if we knew it was better already, we wouldn't have to do the study. And so I think what was really amazing about the OLIE trial is there was real concern from the sponsor, from ASI, that if we randomized a trial to something you could get right off the shelf and not have to be on a clinical trial, and only half the patients got the new drug plus what you could get off the shelf, would patients enroll? You know, and would we be able to finish the study? And you know, the truth of the matter is, they suggested that Dr. Dela Cruz and I come speak to MIB today because they didn't think we would be here by now, which is that yes, patients did join the study because the patients and families want to know just as much, if not more, than those of us who are doing the research, whether or not it works, so we can get it to the beginning sooner if we can. So now we're at the waiting stage of knowing, so all the patients are getting their treatment right now, and we don't know yet what the results are. So when we know what the results are, that will actually, as I said, shine a light on what to do next. But know that in the background, we're already planning this in COG because each of these trials of drugs that are slightly different, but overall similar in their mechanism of action, have consistently shown a positive signal for helping patients with osteosarcoma. So now we just have to figure out how to use them. And that's a hard thing to do. I did notice in the chat, someone said, how do you know which one to give? That's part of the problem, right? They're similar, but they're not. And so some patients will tolerate one drug better than they will tolerate another drug. The most common side effects of these drugs are sort of low-level gastrointestinal side effects. You kind of feel sort of low-grade nauseated all the time, not like those really bad bouts that you get when you got cisplatin, but you sort of feel lousy, you know, food doesn't appeal to you some of the time. Some patients get diarrhea with it. That's probably the most common, and feeling tired. Some of them, sometimes you get very high blood pressure. And if you think about the way that those drugs work at sort of trying to prune blood vessels, it increases the resistance of the part pumping because there are fewer blood vessels to pump into. So when we see the blood pressure go up, that's actually a good sign that the drug is doing what we'd like it to do. So we treat the blood pressure to bring the blood pressure down. We don't go backwards on the dose. We don't backpedal and try to give a lower dose. Some people get rashes. Some people get a sore tongue. And I think the trickiest part that we're going to have to figure out for osteosarcoma is normally your body doesn't need new blood vessels to grow, but there are certain times when you do, like when you're healing a wound. And surgery is such a big part of the treatment of osteosarcoma that this is one of the tricky things that we're going to have to figure out about how to use those drugs. So, so we choose which drug based on, if there's a trial open, we'll always encourage you to go on the trial so that we can all learn something. If not, you know, we may try one because we can get our hands on it. So if they're FDA approved for another indication, we may be able to get it for you. If there's good data, like the data that Dr. Dela Cruz showed, we can usually convince an insurance company to pay for it. And then if one is gives you too many side effects, we may try another. And sometimes actually, because they're slightly different, I think of them like, Anne, you asked me about playing a musical instrument. I actually think about these drugs like chords on a guitar. They're different strings, and some of them press a little bit harder on one string than another, but they're all sort of hitting the same pathways, but with a slightly different emphasis. So one's like a sharp and one's like a flat, and, but they're all kind of playing the same tune. But so that if one doesn't work for you, that doesn't mean that if you don't switch to another, it might work a little better.
So how easy is it to get the necessary cell information from removed tumor tissue?
So in terms of information, like the information that, so it's easier now, and the information I think that we're referring to is the genetic information. So because a lot of these, you know, these targeted agents are very specific and generally sort of work because you have a specific protein that's mutated. And so more and more centers have the capabilities of taking, you know, a piece of the tumor that the surgeon removes from a biopsy or from a planned resection and do sequencing. And there are many different degrees of sequencing, but I think there are, you know, more and more that the ability to do that has become easier. So.
But I would add to that again that it's a little bit of a chicken and an egg with osteosarcoma. Osteosarcoma, the sequencing hasn't really to date told us which treatment will work for you. But when we see patients who have an extraordinary response, we want to have that tumor sequencing so we can say, oh, okay, well, now we know they responded. Let's go back and say, what was different about their tumor versus sort of all the other patients who didn't respond to that, and see if we can figure out what those biomarkers are that will tell us which patients are most likely to respond to which agents. And that's, you know, when we can do that from some of the standardized sequencing that lots of people get from either Foundation One or their individual institutions. But that also leads to, you know, what Anne alluded to earlier about a project that Dr. Dela Cruz and I are working on to try to use computational biology and computer science algorithms and sort of building networks based on everybo's tumor to try to figure out what might work best for your tumor. So stay tuned on that one, because hopefully down the line, again, as we feed more and more information into these computer algorithms, we'll be able to figure out which tumors responded to which drugs and why, and can we predict now based on all of that information which drug might be best for you. So that's, again, all of these clinical trials are really adding to that data that will help us to figure out if we can come up with a predictive tool to figure out what's best for an individual. We're not quite there yet, though. But each time,
it's so hopeful.
I know we're almost, you know, and
the next frontier. So
exactly, and that's, you know, again, no matter what, these trials are building those databases.
Yeah, which is key, and how exciting that it's international. This is really exciting, and hopefully all that data gets quantified and easily shared and understood by everybody. Okay, we are over time. I think we still have questions, but we'll send those to you, and then we'll see if we can get back to everybody. But in the meantime, thank you so much, Dr. Dela Cruz and Dr. Glade Bender, most of all for your commitment to osteosarcoma patients everywhere. Really, really a big deal to us to have really smart people focusing on osteosarcoma. As an osteosarcoma patient and as somebody who loves a lot of osteosarcoma patients who are really suffering, it's a really big deal to us. So more information on this and all OsteoBites can be found on our YouTube channel, on mibagents.org, or wherever you get your podcasts. We've got over 50 now, so you can go back and go through and find out more about these other trials that we've talked about today as well. If you registered for this OsteoBite session, you'll be emailed the links to all of those places shortly. And just want to remind you too that we do need your support on Giving Tuesday. We are committed to $43,000 being raised on Giving Tuesday in honor of the 43 kids who are diagnosed with cancer each day. Have to do better for them. Together we get to cause a cure, and we're going to do it. Until then, thank you again to our brilliant doctors, Dr. Dela Cruz and Dr. Julia Glade Bender, and to our panelist, Anna Eggleton today. Thank you for joining us, and happy Thanksgiving, everyone.
Happy Thanksgiving, and
Happy
thank you
Thanksgiving.
for everything you do.
Thank you.
Yes, thank you.
Subscribe to our YouTube channel.